Hi friends, greeting from Anamol Laboratories Private Limited. In a series of empowering our scientific and medical community, today we will take a most hot topic like coronavirus. There has been a lot of knowledge which has been coming through you, through the media, TV, etc. But let's take today what is a lesser known knowledge or knowledge facts based on this coronavirus. You see this coronavirus, I have written coronavirus in bracket, I have written an S also. S means, it's a misnomer that this is coronavirus. Corona is a family and it's comprised of many types of viruses in that. So it's a coronaviruses. Point by point, let's come down to what we can understand. There are few terms which are described. There are a number of places where they say COVID-19. Some of them say SARS virus. Some of them say CoV-2. Let's understand them one by one. COVID. COVID is coronavirus disease. 19 is the year in which it has been born. So why it is called as COVID-19. Okay, now let's take another term, SARS virus. SARS basically is severe acute respiratory syndrome. Severe, you know, it's an English term. Acute means sudden. Respiratory syndrome. Syndrome is a group of symptoms which you can exhibit in your body when you have a disease. Now let's see CoV-2. Coronavirus 2. Type 2. This is what has been prevalent and causing havoc and pandemic in the country presently. Now let's take a second thing. What is the size of this virus? Just to give an idea, if you take a one human lung cell, why lung cell? Because this is what has been affected mostly. So one human lung cell can accommodate almost 100,000 coronaviruses or 1 lakh coronaviruses. That small is the size of it. So you cannot obviously see this. What is the shape? There are viruses number of types. Helical, capsular, spherical. This coronavirus is a spherical. You see this diagram? You have seen much better diagram on the TV many times. But this spherical virus has spikes. What are these spikes? You can see these blue marks, spikes. And much better photograph. You can see and elaborate on the TV or media anywhere. It's something like a small, the children play a small ball, rubberish ball, which they throw on some smooth surface and the ball gets stuck there itself. That is what is the kind of a structure this coronavirus is having. And literally meaning these spikes really get stuck. We'll come to it, what exactly this sticking is. What is this virus's composition of? ssRNA, single strand RNA. This is an RNA virus. Most of the viruses are RNA viruses. And it has got little amount of protein. Why we are coming to this? Because this information is very important for us to later on design and understand how this disease happens. Single stranded RNA, which is in the center, and the proteins which are on the spikes as well as on the surface of the envelope. Okay. What is the target cell where it acts? Coronavirus or CoV-2 largely attacks on pulmonary epithelium cells. What is the pulmonary epithelium cells? Is the inner lining of the lungs. Okay. Our lungs have a lot of cilia. Cilia only moves in one direction, preventing a reverse flow or any entry of foreign particles. But these coronavirus attack only on that surface, making their function rendering useless. What is the pathology? Before we come to pathology, let's see how these viruses really react. Our pulmonary epithelium, that is lung cell lining, has an enzyme called angiotensin converting enzyme, ACE. So ACE is a good receptor of these spikes. These viruses, once they enter into our lung system or respiratory system, they bind to this ACE enzyme, ACE receptor. Okay, moment they come into the contact, that thereafter their proteins sitting on the molecule of this virus or the structure of this virus opens the lung cell, enters into inside. Now what happens? Little going technical into that, but is more important. This RNA which is there inside, now RNA has a two fate. RNA using messenger RNA technology, it immediately make its own copies. As you know, when your RNA makes a copy, it has a positive RNA, that is the original RNA, and the copy is antisense RNA, that is negative. Negative in the sense it's a complementary sequence of nucleotide. Okay, now both the type of RNAs, sense RNA, that is positive RNA, that is original RNA of the virus, as well as the antisense RNA, both have their function. The sense RNA make more copies and more copies and more copies for its further making an assembly line of its own, and antisense RNA immediately splice into smaller pieces, each using protein mechanism of host cell make more and more proteins. These more and more proteins are responsible for making the entire virus molecule again thereafter. Okay, you understood this? Both the type of RNAs are important for it. But while using this entire mechanism in the pulmonary epithelium cells, it makes the ACE redundant. That means it increases the angiotensin 2. Now angiotensin 2 increasing in the pulmonary cell has its own cascade of reactions. It increases the vascular permeability of the lung. Means the entire lung system is not functioning as normal, it is defective. Cascading it down, it has angiotensin 2 has a reverse reaction towards kidney function, renin, and that renin makes blood pressure high. Now, so this moment you have this infection, your lung function is impaired, at the same time the blood pressure is rising. Okay, after this understanding of this, we'll move down to the next slide and see what is the next available. Yes friends, now we look into what is the immunity. Immunity with respect to corona infection. Immunity largely is the production of antibodies, and these antibodies are of immunoglobulin G type or M type. These are produced in response to infection when the coronavirus invades into our lung system. Okay, these antibodies are produced, especially I would say IgM. IgM is more important because this is an initial response. This antibody is very strong. It has got 5 sites on which it can attack on an antigen and kills it. Later on, it's taken over by IgG, which is a routine antibody with a single prong to attack the antigen. There is another mechanism of interferon. Interferon is something I would like to explain it to further is. Interferon is a substance, the biomolecule. When one cell is infected, it sends a signal. Short nucleic acid molecules, it sends to the neighboring cells, there is an infection with me, please produce proteins to fight. But this virus immediately interferes with your interferons and does not allow the next cell to know. So next cell, when child virus or the son virus attacks the next cell doesn't know, it welcomes it like a guest and gets infected again. So what is the antibody? Immune system is compromised in patients, those who are above 65 or those who are children below 60, their immune system is weak, hence they are the easy target for coronavirus infection. Whereas those who have a strong immunity system, their antibody, moment the virus infection happens, their entire immune system gets geared up, immediately produces IgM type of antibody to fight infection. Thereafter, IgG type of immunoglobulin takes over and fights the infection to finish it. Hence, not all the infections are able to result into a disease. Okay, what is the diagnosis? There are two types of diagnosis possible. One is RT-PCR. Let me explain what is RT-PCR. You must be knowing NAT, nucleic acid test. Nucleic acid test is where reverse transcriptase polymerase chain reaction is the method used to find out the virus. That means what you are doing is you are taking body fluid where there is an infection possible or suspected. You pick up that and break this and release the RNA part of it. The coronavirus's RNA is isolated first, given it to reverse transcriptase where RNA turns into DNA. Then this whole PCR reaction takes over and the number of copies of PCR, number of copies of DNA through the PCR is being made. DNA is a double-stranded. Cyber green dye which comes into the picture immediately fluoresces, and moment it fluoresces, it says that there was an infection of this particular type of virus. Why this is specific? Because there are probes which are specific to viral DNA, viral RNA. Okay. Now there is a second type of test which is going to be introduced over a period of time, which is a screening type of test. This screening type of test is a rapid test. RT-PCR really takes 4 or 5 hours, whereas this rapid test, 15 to 20 minutes, they are able to detect the antibodies which are raised in our body in response to this coronavirus attack. As I said, coronavirus has spike proteins, surface proteins, and there are certain number of proteins which are around the RNA molecule inside. Any of them are being targeted and antibodies are produced against it. They are IgG type or IgM type. So the rapid test has like a pregnancy type of test, has 2 lines, IgG, IgM and control line. Now when there is a suspicion of a person having some kind of a disease similar to corona, then you immediately apply this rapid test. If you find it positive, go through RT-PCR confirmation. Okay. Now let's come down to what is the treatment possible. There are drugs which are targeted towards various parts of this coronavirus. Monoclonal antibodies which fight against various proteins of the virus. There is a chloroquine, which is an anti-malarial drug. This anti-malarial drug has a tendency to increase the pH of lung tissue. And increased pH does not allow messenger RNA of coronavirus to make its function go far. Hence, people taking chloroquine are less prone to infection with the coronavirus. There is a HIV treatment, a combination of drugs. They are good in arresting the viral RNA multiplication. Hence, coronavirus treatment also is possible through HIV set of drugs. Okay. Vaccines. Okay, let's come down to what is a vaccine possible. Let me explain what is a vaccine. Vaccine basically is a pre-prepared antibody response in our system to fight future infection. We can take any part of the virus. There are 30 different candidates right now working on various molecules of virus DNA, virus RNA, virus spike protein, and they are trying to produce antibody line against it. What is vaccine? Vaccine is basically a coronavirus, various parts, but attenuated. Attenuated means they are heated to 56 degrees centigrade for 2 or 3 hours. So it loses its capacity to cause the disease, but does not lose its capacity to produce antibody response or immunity response. So they are targeting spike proteins, they are targeting surface proteins, and some of them are targeting even messenger RNA proteins to inhibit the infection of coronavirus. I hope, friend, this has been successful. Make you understand some terminology and some unknown facts about the coronavirus. If you like it, subscribe it. And if you want to see more of these kind of things, press the bell icon. Thank you very much, friends.